Dr. Daniel is the W.D. Miller Professor and Vice Chair of the School of Dental Medicine at the University of Pennsylvania. He's a AAAS Fellow and a member of the National Academy of Sciences in Italy. He has received numerous awards, and many are related to his pioneering efforts in developing chloroplast genetic engineering concept to allow orally delivered stable protein drugs to treat major metabolic, genetic, and infectious diseases. And so this innovation is being brought to the clinical realm with funding from agencies such as the FDA and the CDC, backed by organizations like the Gates Foundation, NIH, and major pharmaceutical companies. So it is our honor to have Dr. Daniel join us today and share insights in affordable protein drug delivery using plant cells. And he's going to talk about a very relevant topic of RAAS modulation in COVID-19. The stage is yours, Dr. Daniel.
And thank you for the invitation, and really appreciate the previous presenters and excellent scientific innovations. I'm going to present today probably a new way of introducing protein drugs in veterinary medicine, and this is also new in the human clinical trials as well. The rationale is to reduce the cost of protein drugs, and of course the focus will be primarily on the RAAS pathway, with also the impact on SARS-CoV-2 and COVID-19 as previously introduced by the previous speaker. So the major, for the past 50 years, protein drugs have been made in fermentation systems and yeast, E. coli, CHO cells. They are purified and injected. And this increases the cost of protein drugs. And many of the protein drugs are not available to a large majority of the global population. And it is certainly not that much available for veterinary applications as well. So the new approach that we are using is to eliminate the need for fermenters, purification, cold chain, injection, but to produce them instead of microbial system or cell culture system, we clone the genes, express them in plant cells, and they can be orally delivered. I will show you some of the mechanistic aspects of this. So the major goal is to make protein drugs available and affordable. And a couple of other examples other than RAAS is that diabetes is, globally, it's a global epidemic, and there are insulin pumps that cost $6,000, $12,000 a pump, a patient, but 1/3 of the world earn $2 a day. So many of the recent drug delivery innovations and inventions, the majority of the global population is cut out of these benefits. And in addition, injections are definitely not very patient-friendly, and it is worse in certain organs of the body. I teach in the dental school where protein drugs are approved as devices and they are implanted. So it's surgical delivery, and therefore there is pretty low compliance. So our approach is to make them in plant cells so that they could be delivered as capsules, or for topical delivery, they could be delivered even as chewing gum. And I'll give you a couple of these examples. Many of you may have had your plant biology several years ago. So real quick, capturing of the basic techniques and concepts is that the step one is to clone the gene, whether it is ACE2 or insulin, it's all the same thing. The cloned gene is introduced into plant cells using a brutal force. It's a gene gun. This may be one of the few examples where gun can be employed for useful purposes. And you coat these DNA plasmids on gold particles, shoot them in, and then you look for with the selection process, you look for the shoots that capture the gene, and you continue to grow them. And once you get the seeds forever and ever will contain the gene, and they are ported to the pharmaceutical companies that produce plants under CGMP conditions. And then these are freeze-dried and made into capsules. And this is all the processes are meeting FDA requirements. So through this process, what we accomplish is elimination of fermenters, elimination of protein purification, and also the cold chain. Essentially, these freeze-dried powder plant cells are stable for several years, and therefore it saves a lot of cost, and vaccines and pharmaceuticals can be mailed instead of injection in the clinic. How many proteins have this approach been tested? Over 200 proteins. We have done a lot of infectious diseases, from bacterial, viral, protozoan pathogens to ACE2, angiotensin, angiotensin, blood clotting factors, main basic protein for Alzheimer's, growth hormones, and so on. So this basic concept is that clone the gene, use a selectable marker, but then the past few years, we have removed the selectable marker before we take them to the greenhouse. And so ultimately, the final product doesn't have the antibiotic-resistant gene. I'm going to show you the video of, it might be a little bit noisy. This video is taken in the commercial facility, so I will not talk as this video is played. Conditions that doesn't have as large a capacity as the one that is shown here. This is aeroforms. Aeroforms produce supplies lettuce for several of the eastern United States. We used to get them from Salinas Valley, California, and transported 4,000 miles here. But now most of the lettuce is grown indoors, and therefore the ACE2 has been produced in both Fraunhofer and the Airform facilities. So we have the ability to scale up. And according to FDA requirements, we should measure the moisture content. So we look at the drug dose, antigen quantitation. We also look at the fresh weight and the the moisture content, because the weight when the capsules and gums are made based on the plant powder weight, the dose is affected if moisture content is different. Therefore, we have cutting edge freeze drying machines with FDA regulatory requirements for self-cleaning and bioburden, all the other evaluations. So we produce products that is ready for the clinic. As you can see, Fraunhofer, we have worked with them for several years. Therefore, the drug expression level is almost 10 times higher than aeroforms. Aeroforms started producing this only for the first time here. So we need to do more optimization and lighting conditions and other things. So that is step one, how to produce proteins in plant cells, eliminating the cold chain and eliminating the need for purification and injection. The next major step is it possible to orally deliver them? So there are two major obstacles for delivering proteins orally. First, if insulin is orally delivered, it will get digested in acids and the enzymes in the stomach. So when you express it inside a plant cell wall, the plant cell wall is made up of polymers with beta linkage. Human enzymes break down only alpha linkages. Therefore, plant cells protect the proteins orally delivered through the stomach. And as you can see in the left bottom panel, the PC is plant cells with GFP. You can see that GFP is fully protected. But then the next step is how to get it from the plant cell into the gut epithelium. Here, we rely on the natural system, which is gut microbes. The gut microbes produce enzymes that break down the beta linkages. So when they break down the beta linkages, in this case, GFP is released into the gut lumen, and it is then absorbed by the epithelial cells. Of course, this doesn't happen naturally, so we have to use proteins to get them across the gut epithelium. In the right top panel, you see different types of transmucosal carriers. We use either receptor binding or non-receptor binding. In other words, these TTB binds to GM1, which is a universal receptor, and it's very, very efficient. And we can also target them to specific tissues, to the immune system or non-immune system, so we can target them to the circulation or to the immune system for vaccination as well. So this has been demonstrated for a large number of proteins, and here is a review that you can look at to understand this. So one of the first applications of this were from pharma companies want to decrease the drug allergy for the protein drugs that they deliver. One of the such common challenge is the blood clotting factors, and blood clotting factors develop antibodies, and by delivering blood clotting factors orally, we could do the immune suppression. That's the natural function of the gut immune system. And we have used this. Last year, FDA approved the RAAS protein made in peanut cells to suppress peanut allergy. So gut immune system is quite a powerful system to block allergies created by proteins drugs. This is applicable for egg allergy, milk allergy, and pollen allergy, and so on. And so we have developed this for over 10 years. As you can see, a number of publications, some of them featured on the cover of Blood, shows the importance of the application of this approach. I'll just give a couple of examples before getting into RAAS. So this one is the human factor 9. If you inject factor 9 to hemophilia B mice, the allergy is so much that anaphylaxis, most animals die. But when you deliver this orally, you could block the anaphylaxis and block this. This we have done in dogs as well, hemophilia dogs. We have finished this almost a year of study with the University of North Carolina dog colony, Beagle dogs. These are 25-kilogram dogs, where, as you can see, the dogs were fed with lettuce orally. They loved it when we mixed the bacon flavor with the plant cells, and they were looking forward to feeding. So we fed dogs twice a week, and these studies have been done for 3 months to 1 year. And as you can see, the dogs that didn't get the drug had very high antibody titers, and we have to give Benadryl to decrease the anaphylaxis and allergy. And this is true with oral insulin also, and oral Biorad. So pretty much small protein, large protein can be delivered orally. We compare the injection versus the oral. And we have also demonstrated this for long-term studies like insulin like growth factor with bone regeneration in diabetic fracture healing as well. If you want to learn more about this, these are the publications are listed here. And since this is a RAAS, I just wanted to give you a brief introduction of the breadth of this technology, but then get more directly into the RAAS system. With so many excellent speakers so far, me introducing this topic is like bringing coal to Newcastle, so I wouldn't invest too much time on this, except to point out that most of the clinical drugs are inhibitors, and that decrease the negative impact of the pathway, the RAAS downregulation pathway, but then the positive impact. There are not many drugs. The previous speaker pointed out about one example for treating the COVID-19 patient. That's only one patient, and that is the ACE2 enzyme. So there is a lot of opportunity for a combination drug of inhibitors in addition to enhancers. The enhancers is the promoting the positive aspect of the RAAS regulatory pathway. And so the first question is, can we deliver ACE2 and Angiotensin 7 in a dose-dependent manner? As you can see here, these have been done in a number of different groups. And the left panel is done in University of Florida, right panel done at University of Indiana, and my lab. So it is possible to do in a dose-dependent manner. This means that there is enough protein is protected in the stomach, and when they get to the gut, there is no limitation of the gut microbiome. There is enough gut microbes to break down the plant cell wall, and then there is no saturation of the receptors, the GM1 receptors. So all of this is functioning in a dose-dependent manner. One thing I want to call your attention to, compared to the previous speaker, is that the previous examples done with ACE2 was done with truncated ACE2. So the commercially available are the one that's the one human clinical testing was done. The ACE2 was made, but ACE2 is membrane associated. It's difficult to make the protein with the transmembrane domain. So that Zufle publication used a truncated protein where ACE2 transmembrane domain is deleted. But we decided to make the full length ACE2 because that's what is in humans and that's what in animals as well. So our ACE2, when we orally deliver, even though I'm showing here plasma levels, in fact, when we deliver ACE2 and Angiotensin 7, there is a 10-fold increase in the lungs. So because we have transmembrane association, we are able to deliver this ACE2 to the membranes and also to the plasma. So that is a pretty important distinction I just wanted to make from other previous studies that have been done with the ACE2. So they refer to the ACE2 as soluble. We refer to as full-length ACE2. And again, this is published in Biomaterials a few months ago. We have used a number of different systems to evaluate the RAAS and oral delivery of ACE2. The most common system we use is the delivery of the disease model. We use monocrotaline to induce pulmonary arterial hypertension. So with the pulmonary arterial hypertension drug-induced monocrotaline-induced model, we could evaluate this to look at a number of different parameters: suppression of antifibrotic effectors. As you can see, TNF-alpha, you could see the controls versus the treated, and you could see a pretty significant reduction in fibrosis. Also, the collagen staining was evaluated to the ventricular fibrosis. Again, you could see a pretty significant reduction. This is after 2 weeks of oral delivery of ACE2 and angiotensin. And when you look at the pulmonary vascular remodeling, we look at the medial wall thickness, you can see a pretty dramatic increase in reduction. And systolic pressure, right ventricular systolic pressure, is also restored to significant levels. So a number of different parameters are used. More direct evaluation of this is through measuring the pumping rate. As you can see here, the right ventricle is enlarged, and in monocrotaline, when we do the combo therapy, in all of these things, combination of ACE2 and angiotensin worked much better than either alone. And as you can see here, we restored the pumping capacity to 95% in 2 weeks of oral delivery of ACE2 and angiotensin. And so our model is to deliver both at the same time instead of either angiotensin or ACE2. And we have quantified this after 2 weeks of the combo treatment. We have measured the area of right ventricle. We have looked at the pulmonary vascular resistance, right ventricular hypertrophy, and most of this restoration is quite significant and reproducible, performed with different groups across the country in the US. And therefore, Dr. Mark Oyama, who is at Penn Med School, started the studies in 12 dogs, the initial round of study. This is the 5 days treatment, 5 days washout, and the dose that we calculated was based on the dose that we observed, efficacy dose that was observed in monocrotaline treated rats. So here we use the 800 micrograms per kilogram and of ACE2 and 0.2 milligram per kilogram of angiotensin, and blood samples are collected, and we are in the process of looking at it. And there was a comment about the collection of the blood samples. One of the things that we found was that when we use EDTA in the collection, then we are unable to measure ACE2 activity in plasma. So it's important to avoid EDTA in the collection tubes, and so heparin tubes work fine for the collection. So initial collection was with EDTA, but subsequent collections were done with heparin. More recently, because of COVID and limited number of pets at Penn Med School, Mark WAMA has developed collaborations with Dr. Jessica Ward and Dr. Marissa Ames. They are all, I believe samples have been shipped to them and they are going to be initiating the study. I received an NIH grant to continue this for the next 5 years. It's a R1 grant, and therefore these studies are already funded. So here the goal is to do this for 21 days of oral treatment and then look at the ratio of alternative to the traditional angiotensin peptides, to look at the tolerance, biomarkers, renal function, and echocardiographic heart size measurement, just as I showed. And just as any other study, it takes more than 10 years to get to where we are today. For example, for the hemophilia studies, we published 10 years ago in hemophilia mice, then we did it in hemophilia dogs, and we have completed with the NIH SMART funding, toxicology and pharmacokinetic studies at Stanford, and we have filed INDA. So it's a long, slow process, but all the single dose, multiple dose pharmacokinetics have all been completed with this. So we have, but when we submitted these, the same studies were done with toxicology and pharmacokinetic studies at phase 2 and Angio17. Unfortunately, FDA didn't accept our studies for IND filing. They said that the Stanford study was only for 8 days. We got squeaky clean results. They didn't like it. They really wanted to see some toxicity when phase 2 and Angio17 is orally delivered. So they asked us to do 24-day oral and 14-day and 14-day post-dosing to observe. And we have now, because Stanford Research Institute was closed for COVID, we used Charles River Labs to repeat these studies, and these studies came out pretty clean, no toxicity observed, and now we are submitting this to FDA in order to proceed with human clinical trials. So at this stage, I'm switching gears to look at, to present the data, the more exciting data that we have, which will have quite a bit of relevance to the discussions you had after the last presentation and the connection between SARS-CoV-2 and the RAAS pathway. So ACE2 is the binding site for SARS-CoV-2 entry into the lungs. Angiotensin 2 is pro-inflammatory, pro-thrombotic. Downregulation of ACE2 happens, and therefore increasing ACE2 and Angio17, excess Angio2 leads to acute lung and cardiac damage, which is shown in a lot of studies. This is the CDC data showing how the heart disease and how the blood pressure plays a key role in the fatality of COVID-19. And the concept here is that there are multiple interactions. First is that the enzyme, the SARS-CoV-2 produces the spike protein, which is shown in red spikes here. So the protein directly binds to soluble ACE2 and blocks the activity. And at the same time, I will show you data that the ACE2 activity dramatically decreases in COVID-19 patients, but not the ACE2 concentration. In fact, the ACE2 concentration decreases. And there are also two receptors, and we, being in the dental school, we use the oral system, and in the oral epithelial cells, just like lung epithelial cells, it's ACE2 receptor, but there's also a co-receptor. In fact, there is two co-receptors for each ACE2 receptor, which is a GM1. And So this is our concept for using the ACE2 fused with CTV to block the entry of SARS-CoV-2 through the human epithelial cells. As I mentioned, the spike protein binds to the ACE2 directly. In fact, you see here soluble ACE2, monomer, dimer, trimer, all of them are able to bind and effectively block the ACE2 activity. The ACE2 receptor is widely written up, but there's not much written up on GM1. Indeed, if you look at the spike protein, the RBD binds to ACE2, but then there are two adjacent GM1 sites. So you need both receptors. Without co-receptor, ACE2 alone cannot facilitate entry. So we use this concept to study the trapping of the virus. So we express the ACE2 in plant cells. We made the chewing gum, and we collect patient saliva or patient swab samples, and we look at the ability of the gum powder to block the trap and block the SARS-CoV entry. So one of the things to study entry we used was use a virus system. This is a lentivirus system. And in the lentivirus system, you express the SARS-CoV spike protein on the surface, and then you incubate this with vero cells. As you can see in the control, they are able to enter the vero cells. The spike protein binds to the receptor, ACE2 receptor, and enters. But then when you add the gum, the placebo gum has a little bit effect because there is some sugar alcohols in the gum. So, and kind of ironically, if people during the epidemic, if they were chewing gums, they may have had some protection, but not a whole lot. But then if you add ACE2 to it, it pretty much blocks. And the exciting things here is these were done with very high viral titers. And with 50 milligrams, we are able to achieve 100% blocking of entry. But each gum has 2 grams. So this is a pretty significant blockage. And we did the same thing with VSV. Here, this is with the, I will acknowledge all the participants at the end. This is also with the vet school here with on Hardy's lab, where VSV engineered with spike protein enters the vero cells. But then if we incubate it with the gum, ACE2 gum, it completely blocks the entry. So we have shown that we could use the ACE2 gum to block the entry. But can we trap the virus? So here is a new method that was developed here at Penn, where we could visualize the virus in the saliva or in the swab samples using a cell phone technology and microbubble technology. Essentially, the antigen, the viral antigen, N antigen, the antibody is added, it traps, and then it develops the oxygen bubble, and the size of the bubble is proportionate to the virus size, the virus particle size, and also the number. So if you look at this, these are different patients with the microbubble evaluated. As you can see, it's a lot of virus in the patient saliva samples and swab samples. In some of them, the virus, the particle size is huge because these are more advanced patients with much larger particles. And when we treated, of course, with the placebo gum versus the ACE2 gum, you could pretty much greater than 95% wipe out the virus particles. So what it does is if you decrease the virus particles, 2 big advantages. It gives the patient to develop immunity because it's not reinfecting the patient, but at the same time, it actually blocks transmission. It helps the patient as well as it helps the neighbor as well. So this is again another example. So when we have done a lot of different samples, as you can see, it's completely wiped out. So we use ACE2, which is a RAAS pathway enzyme, either to trap the virus or to block the entry. Two different mechanisms. And we could even do this with PCR. We could, because we thought that PCR is quite sensitive, and but we could detect it using the PCR as well. So essentially, the gum is 50 milligram testing here, but then actual gum used in tested in patient is 2 grams. And we are also looking at this concept to debulk other common seasonal flu virus and so on. So another interesting aspect of the COVID-19 and the RAAS pathway is that these are human plasma samples. As you can see, when you measure the ACE2 activity, there is a dramatic drop in ACE2 activity in COVID patients if you look at the panel A. But when you measure the ACE2 concentration, there is a dramatic, the ACE2 concentration in COVID goes up. So if someone is measuring just the ACE2 quantity in the plasma, it might give a different interpretation than ACE2 activity. And how does this, how did we investigate this further? We measured Angiotensin concentration. As you can see in the control, except 1 or 2 patients, the Angiotensin concentration is pretty good in control patients, but in COVID, it is pretty. So you look at the enzyme, you look at the product, but then the enzyme concentration doesn't correlate. So there is a lack of correlation, but there is of concentration, but there is a correlation with ACE2 activity. In fact, I have not shown the slides here. We looked at the membrane-associated ACE2 in lots of lung autopsy samples as well. And there we didn't see between the control and COVID patients the concentration of membrane-associated ACE2. So the challenge in doing plasma, again, there were some questions about inhibitors. I'm not sure that was protease inhibitor or ACE2 inhibitor. The problem in measuring ACE2 activity is that there is inhibitor, and therefore we have to remove the inhibitor before we measure the ACE2 activity. This is what a lot of groups miss, because when they measure the ACE2 activity without removing the ACE inhibitors, then they get misleading results. But the good news is that when we measure ACE2 activity in the saliva, there is no need for EDTA collection, there is no need for removal of ACE2 inhibitors, and there is no need for cold storage. You can store saliva at room temperature. So a lot of the problems in evaluation of ACE2 RAAS pathway correlation can be eliminated by using saliva. In fact, this manuscript is in review now. This will be the first published report of measuring ACE2 activity in the saliva. We looked at the literature. We really couldn't find any. And so in panel A, you can see that the red horizontal bars are the COVID-19 patient saliva. The ACE2 activity is completely knocked out, so it gives the virus free passage to enter into the oral epithelial cells. It has no control. And whereas in healthy patients, the ACE2 activity, the ACE2 can block in 2 ways, either by binding directly to the spike protein or binding to the ACE2 receptor as well. One interesting finding here is that these are samples from Penn hospitals. So we found one COVID-19 PCR positive patient, and then we found ACE2 activity. When I called the hospital, and said that probably this person may be asymptomatic, they got very upset. They said, we don't admit people, asymptomatic people in the hospital. But then a few hours later, they called back and apologized and said, sorry, yes, this patient was asymptomatic. There was no COVID symptoms. So it's interesting that saliva ACE2 activity is a better predictor of SARS-CoV-2 infection than the PCR. And so this is ACE2 in saliva could be a good biomarker that could be developed, and so RAAS pathway could contribute to that. There was also a question about how ACE2 decreases in the previous talk. So here we show that direct binding of ACE2, spike protein to ACE2, we have looked at the full-length spike, we looked at RBD, it completely knocks off ACE2 activity. We also looked at Western blots and combination and so on. So there is a pretty direct impact of spike protein binding to ACE2 and blocking. However, we don't rule out the downregulation of ACE2 as well. So there is a downregulation of ACE2 as well, because we are not saying that in the plasma all of the decrease in ACE2 activity is related to the spike protein, because there is not enough data available about the concentration of the spike protein in plasma or saliva. That's something that we are investigating. So based on the data that I presented so far, we have, with the clinical team here, we have developed the clinical trial, we have submitted the IND, and both in the hospital and outpatients as well. Of course, the chewing gum will be done in outpatients because it's needed only for a 30-minute chewing to measure the ACE2 before and after the chewing gum. Whereas for COVID-19 or any other heart disease treatments subsequently, this will be a 2-week treatment. That's what we have done so far. And as far as oral delivery regulatory pathway, the peanut allergy RAAH has been approved last year. So oral delivery of proteins made in plant cells, the regulatory pathway is already available. So with this, I will make the not all the conclusions related to RAAS pathway alone, but also I would like to introduce this to your group, that this oral drug delivery is affordable because it eliminates a lot of expensive purification, fermentation, cold storage, and so on. And edible plant cells can be used to produce them, and it can be used to treat infectious as metabolic diseases. And protein drugs are stable in freeze-dried cells for a number of years, and they can be orally delivered in a dose-dependent manner, as I mentioned to you, gut microbiome, and also the transmucosal carrier, both are needed for that. And the commercial facilities are available. I showed you a video of Fraunhofer, and the IND has been filed to treat these initially COVID patients, but eventually we want to continue that for the heart disease and the hypertension as well. So in a sense, this offers veterinarians a reasonable new approach to deliver drugs in veterinary medicine, especially protein drugs, in combination with the small molecule drugs that is currently in use. I want to acknowledge all the funding that we have received from a number of NIH funding, especially for the NHLBI funding, and the SMART program paid for the toxicology, and a number of different the pharmaceutical companies as well. I also want to thank American Heart Association for highlighting this and featuring it, and giving the best innovation award as well. Want to thank all my collaborators, lab colleagues, and the IND regulatory team and the evaluation, saliva collection, swab collection, all these physicians. A lot of this is done in collaboration with several the schools here. So I will stop and will be glad to answer more questions.
Thank you very much, Dr. Daniel. We'll just give you a silent round of applause. That was fascinating. And we have several questions, and one pertains to the chewing gum and blocking the receptor by which COVID gains access to the oral epithelial cells. Mm-hmm. Is that protection conferred while a patient is chewing the gum, or do we know the duration that it can protect them, or at least prevent COVID from entering? How long does it last?
It's a very good question. As you can see, I'm in the dental school, so the dental healthcare workers want to protect themselves for the 1 hour that the patient is in. So one of the primary applications and goals is to give it to the patient before they sit in the chair. And so the oral the procedures generate a lot of aerosol. So when they have millions of virus particles, it contaminates the whole room, and therefore decreasing it to fewer goes a long way in decreasing that. And this would be true with people eating, chatting after eating food in restaurants, and close proximity in public places and transportation, duration of transportation. But most importantly, it also protects the patient, because it gives patient, if they chew multiple times, they can keep reinfection. So it definitely decreases the virus load and protects the patient, very different from mask, which only prevents the transmission, but not the patient. So here, the duration depends on how long our clinical, right now, once we get our IND approved, we are going to do this in the clinic. Right now, we know that just 10-15 minutes blocks out quite a bit, and therefore the duration of the clinical study would be 30 minutes, but we are going to do 2-3 times to really see. But we do not make a claim that this makes the patient free of the disease. If they want that, they should take oral capsules of ACE2 angiotensin 7. So that is the 2-week treatment where they could fix the RAAS dysregulation. So that is a long term where we claim that the heart and all the other parameters that I showed can be restored during the 2-week treatment, but not the chewing up. So these are, one is transient, repetitive, the other one is long term.
Very cool. And in terms of delivering these drugs orally, what is the difference or is there any indication that giving them to fed animals or people versus fasted, is one better than the other?
Yes, so if you are referring to the RAAS specifically, then that's what Mark is doing, and you're all teaming up to together collect that data. But orally delivered, we have done it for the hemophilia dog that I said, that I showed. That is pretty dramatic difference. That the dogs, they didn't eat lettuce. They didn't like to eat lettuce. They liked the flavor that we put with it, the bacon flavor. But they liked it, and we had no problem feeding. And those studies were done for a duration of a year. We looked at blood chemistry, toxicology, and everything else. There, we were able to maintain the tolerance for more than a year. So there is a long-term restoration by orally delivered drug. And in this case, we are being cautious. We could have done just ACE2 or angiotensin 7 alone, but in rodent studies, repetitively we see a combination effect much better than either one alone. So it shows that continuous maintenance of H2 and angiotensin 7 is helpful on either alone. Did I answer your question?
Yes, thank you. And so we'll invite the audience, if you have any questions in this fascinating topic, please submit them to the chat or the Q&A. And just, I think it's your research is innovative and very interesting. How did you enter this world of plant cell encapsulation and drug delivery?
Yeah, thank you for that question. You know, I grew up in a developing country before I came here. And even today, after being in the US for more than 40 years, still insulin is not affordable in most of the developing world. And there is so much black market, there is bogus insulin and so on, largely because it's just the current system is just too expensive. These fermenters are $500 to $900 million, and the insulin shelf life is very short. And so I was really committed to finding a new way of making protein drugs. And this is also true. I have seen a lot of small coffins in the church and always question how do so many people die. And when I realized that they could afford the vaccines and they couldn't afford. So, even today, look at 5% of the global population is vaccinated. 95% of COVID in the rest of the world is not vaccinated. So this problem is never ending. And so I am really committed to making a better way of making protein drugs available. Small molecules are not this bad. The small molecule drugs are available in India, but the protein drugs are not available in India, and most of the countries are located in Europe or US. And so the goal is to find a convenient way of delivery. I didn't know until I came to the dental school that protein drugs are delivered surgically, and so far injection is the only way to deliver protein drugs, are surgical. And so this is something that we are trying to address, and we are almost close to the finish line.
It's a noble effort. And I think we may have a question. So we have a question: how would gastrointestinal disease, dysbiosis, antibiotics, etc., how does that affect the GI microbiota? So how do GI diseases and antibiotics influence the availability of your delivery system, if at all?
It does. Excellent question. Actually, we have done uptake studies in mice grown in sterile facilities with antibiotic treatment. And when we do the same GFP delivery slide that I showed, we cannot see GFP absorbed in the circulation. So clearly, when the gut microbes are wiped out, then there is no breakdown of plant cells. And therefore, that if somebody is taking an antibiotic treatment that decreases the gut microbiome, it depends on, when we did this experiment, we went to max. We just completely wiped out. It's possible that doesn't happen in patients to a complete wiping out. In fact, people taking antibiotics, sometimes they have their digestive system affected by antibiotic as well. And but then the microbe is restored when they finish the antibiotic treatment. And therefore, yes, so that is one other line of evidence for us to show that our mechanism of delivery depends on the gut microbiome. And we are also using this to treat, we have made interleukin, and we are trying to treat IBD, Crohn's disease. as well. We are also using the system to deliver the drugs for gut pathogens. And so we are doing direct gut delivery as well. There we don't need the transmucosal carrier. So we are using this approach to treat gut diseases as well.
That is an excellent question. Thank you, Dr. Bartges. And unless there's any more questions, we'd like to thank you again. So another huge silent round of applause. Again, it's noble work and fascinating work. And I look forward to continuing to collaborate with you and Dr. Oyama and Dr. Ward.